I'm going to welcome Owen Powell. He has been latent within many presentations in the last day, and I think now it's a nice time to give him the floor.
Thanks, Jonathan. And thanks for the opportunity to speak. I organised one of these symposium a few years ago, so nice to be back as a speaker. So I thought I'd start with a bit of motivation why I'm interested in G/E/M, and I've got a few statements here which start sort of fairly agreeable to everyone and move more towards opinion the lower you go. But I'm interested in more of accelerating long-term genetic gain of breeding programs, and these programs and the traits we're selecting on are non-linear systems. And in breeding, we are aiming to improve something, genetic merit, which often we're thinking in a linear way. And this is often described in an infinitesimal view of quantitative genetics, of genetic architecture. And as we can see on the right, if we think about the whole system, we have the aim of improving the reference population of genotypes over time with this delta G. That's what we want to do, but there's many processes involved in this. So what I'm sort of arguing, and there's evidence for, is that the intellectual view in terms of viewing traits, but also in how we build prediction models, has worked well for short-term or highly correlated prediction problems. But I think there's areas to improve as we're trying to forecast maybe further in time or further in space. So that could be environmental or management dimensions. And from the talks this morning, I think others would agree that sort of status of biological knowledge that informs a lot of the traits we're interested in is at a point where we can think about how do we incorporate multiple sources of this information into prediction models that get back to genetics, but may allow other questions or other opportunities to think about. So as a cartoon view of this, what I've got here is how would we think about going about this. So on the left, I've got a caricature of a infinitesimal model where the small dots on the left are genomic variants, so our sequence information that we might be collecting in a breeding program, and we'll have the large dots on the right, which is our maybe an endpoint trait, maybe across multiple environments or two independent traits. And there's been very many statistical attempts to model these relationships and also how different parts may interact. But what I've been working on, and as a group we've been working on, is how do we capture or bridge information in that gene to phenotype map that Epp was talking about in the first talk this morning. And this is a continuum, and there could be in many forms, but what it could really think about is how do we capture new traits and how they interact, and also a dimension of time. So we can move from the far left that I talked about, continuing to capture more and more complexity as we move forward. Why might we want to do that? If we look at the contrasting views of a crop breeding program, we might have the breeder's view, which I think has been talked a lot about in the conference so far, but we also have an agronomist view and a farmer's view, and they're all getting different looks at this aspect of the end goal that we're really talking about in this G/E/M conference. So the opportunities can benefit all of them. I think ones we think about and share a lot about how do we rank varieties, so looking at the selection accuracy for the breeder's view, that can be thinking about, and then optimization of their decisions. But another one linked to the last talk is, can we evaluate more genotype by management scenarios? So that might be a question that we might need to consider with changes of farming systems that are going to be far more punctuated changes to the system than a gradual climactic change we might want to think about in terms of G/E. And a little prelude, so some of the modeling we've been working on to connect genomics and crop growth models into a platform. We can evaluate many, many combinations of these that you might not be able to test in a field. So we sort of recently just did 16 billion G/E/M combinations going from the genomic sequence through intermediary traits out to complex traits like yield. So I was a bit vague on exactly how you might do this, but what I'm really going to focus on today is integrating crop growth models with genomic prediction. We've heard a few different flavors around that in terms of how we might get QTL information, but this has sort of been done before, so I'm going to walk with a little bit through that here. So this has been a few papers started in private industry working on corn, where they've actually integrated a maize crop growth model with genomic prediction to have all of that information available, and comparing this against a standard genomic prediction model that doesn't capture changes in the environment. And here we have on the dotted diagonal is the one-to-one comparison of a Pearson correlation in ranking these genotypes. And the majority of the prediction samples are above this line, showing a benefit of having this intermediary dynamic information in predicting that particular problem. This was one example, and it can show that you can be optimizing for a single trait or a multi-trait, and that's relatively flexible to do. And also that, with other papers, have showed that it's not just specific to one crop model. I think generally there's sort of been 3 or 4 crop models where this sort of type of improvement's been demonstrated before. For the keen-eyed, I'm looking to the right here, you do notice some dots that fall below that line. And that's something that a lot of people working in this space are becoming aware of, where when you add more complexity into the models, this optimization problem can be a bit more difficult when you can have many solutions matching your observed data set. So that's something that, as a group, as a challenge that needs to be considered and an open question. So the opportunities for what we wanted to work on was, say, it worked for industry in the US. Can it work for a different crop, and can it work in a different setting? So being in Australia, I was working with Mark Cooper and a few in the audience here. So in the Centre of Excellence, what we were looking at is using this hierarchical genomic prediction model. And on the left, I'll walk through the bullet points before going through the more detail. But what we did is we connected the APSIM crop growth modeling platform with genomic prediction. So that also allows multiple crops to be interchanged and parallelized computing to make this efficient and runnable. But really what the point is it's trying to do genomic prediction to these intermediary traits that are generally believed to be more heritable, so more static across those environmental and management contexts. And in this framework where you don't actually have to observe those phenotypes, so they can be modeled as latent variables. So therefore, although they might be quite low in the biological hierarchy, we don't have that additional measuring constraint. And then once we get these inputs into the crop growth model through genomic prediction, we can then pass them into the crop model, and we can allow the functions in the crop model to determine the yield as the emergent interactions. So that's how we're capturing the statistical G by E by M that we often talk about. And it could be starting with crop growth models, but I think in terms of a more prediction algorithm development, it could be extended into different more molecular or lower order biological networks. And maybe a point of distinction to a number of models that are being published in this space is that we're actually, by modeling these latent variables in a Bayesian process, we're capturing uncertainty right from the genomic sequence to the endpoint traits and in between, rather than having to carry through error from estimation of QTL effects, etc. On the right, this is where the workflow is. So to get the process really going is you need to specify a prior distribution for parameters of the crop growth model that you're actually trying to model latently. This will allow you to estimate your marker effects. And as we said, pushed into the crop growth model. And it's going to do this across the full environmental dimensions you define and give you some sort of measurement of error to decide whether this is a better or a worse sample across that multi-trait space. And key for conversations yesterday, you'll get posterior distributions of those intermediary and complex traits, as well as genomic effect estimates or marker effects, which breeders will be using in their programs currently. So the way we wanted to evaluate this was that we generated a simulated sorghum data set by connecting AlphaSimR with APSIM. And this allowed us to give a very specific data set that contrasted for environmental dimensions relevant for Australia. So the 3 traits that we wanted to vary in this population were maturity, a parameter related to canopy development or leaf size, and the ability for a sorghum genotype to tiller. There's genomic data there, including the causal QTL information, like positions and effect sizes. And we generated these sort of fit a large number of genotypes within a biparental population. And there's sort of 12 trials, so it's 2 locations by 2 years, with a three management factors in terms of population density. And then we'd have also yield and biomass, as well as some longitudinal information. So on the right, the main points is that you can see is that we've got very contrasting years. And this is really driven by in-season rainfall due to the cropping system. But you also see between the different colors and distributions, re-ranking of the distributions due to management. So talking about the discussion earlier, we knew this would be a challenging problem to predict against. And that was why we sort of just constructed this initial data set in that way. And it's sort of publicly available for other people to use. So some descriptions to see whether this might be of interest to you. What I've presented is just from a simple variance component model, the different variance component estimates. So we have a positive and significant genetic main effect, but the G by E by M is significantly bigger. So in short, it's not Switzerland. Sorry, Lucas. You also have genotype information, and the plot down on the right is some work I was doing actually with Martin Bohr just last week. And this is showing across the management context how yield maturity relationships are shifting quite drastically and can be caught in intermediary things which are going to be hard to capture with linear models. So just as a sample of some of the work or where we're up to, just want to compare our sort of hierarchical model where we can model maturity, leaf size, propensity to tiller to predict yield versus a sort of pretty standard linear mixed model where we have sort of a fixed trial and a random G by E term, including the markers. The data set, I took just one slice of this. So this is sort of for those 4 site year combinations where there's a range of heritability between 0.2 and 0.6. And what I've done is I split the data set up into sort of clear model fit where we have sort of training and environments and training genotypes, and where we can predict into new genotypes, new environments, or both simultaneously. So just a sort of very quick view of this. Similar view of the graph from before. So on the y-axis is where we have genomic prediction with a crop model. And on the x-axis is where we're trying to use a linear mixed model to capture this correlation between these environments. And the color is just based on the particular trial. So in the top left is model fit, where you see they can perform quite similarly for a group of those trials. There's a couple where we get between a 0.1 to 0.2 predictive accuracy benefit. If we go down to the bottom left, so this is where we're predicting new genotypes within the same observed environments. That seems to give us a slight improvement still. And although the model that we're comparing against can't adjust for environments, the 2 boxes on the left are really to demonstrate that this having the crop model, the predictive accuracy doesn't seem to be decaying. So we're still able to capture this environmental adjustments between some quite contrasting dry and wet years in the data set. That's the timer. Yeah, all good. So, and just to show that this also persists across phenotyping quality. So this, we're modeling against yield alone in this situation. So we could add different levels of noise to genetic signal on those plot measurements. And across that continuum of plot errors, this predictive accuracy seems to hold. So even if you're having relatively noisy phenotypes on yield, it's not affecting the crop growth model worse than regular models. I'll skip that one for now. So we have presented this prediction framework that allows us to capture biological knowledge of these dynamic networks, can access multiple crops, and we're starting to look at more applications with some partners. And I'll just point out Mel Wilkinson, who's here from UQ, if you want to talk about, connect with her. Here I showed some better ranking of genotypes, which would be useful for breeders across diverse environments, lower prediction error, and we're exploring potential to move beyond crop models down into molecular networks that are going to be presented in other sessions. I'll leave it for time for the rest of them. Thank you.
Thank you, Owen. Due time, I'm going to ask to keep the questions for the discussion session.